All right, I think we have a critical mass, so let's go to the next slide and get started. My name's Malia Becker. I use she, her pronouns, and I'm based in Minneapolis, Minnesota on Dakota land. I'm an organizer with the Center for Biological Diversity. We're a national conservation nonprofit, and we work with a combination of the law, the legal system, science, outreach, and creative media to protect wildlife and wildlife habitat. And my role specifically is to draw connections between upstream or root causes of the wildlife extinction crisis and the impact on wildlife and human activity, so human-related causes. Next slide. And so to ground us in this moment, we are currently experiencing the planet's sixth mass extinction. And plants and animals are disappearing at their fastest rate since the dinosaurs, with over 1 million species at risk of extinction in the coming decades if no changes to the current trends occur. And we know that this time the cause isn't geologic, it's not cosmic, but it is human-related. It's human-related activity. So how many of us there are, how much we're consuming, and this idea of endless growth, that we have infinite resources to continue to consume more water, continue to develop more land for human uses, is all contributing to further burning of fossil fuels and a warming of our climate, as well as ultimately pushing out species like the American burying beetle. And I apologize, I don't know if you can hear our monthly tornado siren is going off in the background. It's every Wednesday at 1 p.m. here, so hopefully you all can't hear that. So over the past 50 years, the human population has more than doubled, while wildlife populations have plummeted by an average of 70%. And that is concerning. So next slide. And here in the US, we are a very high consumer, high wealth country. So the US is 5% of the world's population, but we consume 30% of the planet's resources, and we create 30% of the planet's waste. And this is significant because even though the US has a low fertility rate, we have each child born here has an outsized impact on the planet due to our high consumption rate and our high consumption economy. Next slide. So all of that is background to these two projects at the center that I want to touch on briefly. The first is our Endangered Species Condoms Project. So this is a free condom distribution program that really works to expand conversations and open up conversations at this intersection of human activity and the impact on wildlife. So you can see in the center photo here, these are boxes, and each box has an endangered species on it, one of these puns, and then on the inside there are 2 condoms and quite a bit of information about how that species is impacted by human development or growing human activity. And this is really to sort of break down taboos and open up these conversations about potential rights-based solutions. So things like universal access to sex education, access to contraception, and reproductive rights generally, as well as voluntary family planning. And then over here on the right is our Protectors art exhibit. This is a silkscreen exhibit that really showcases the protectors or stewards, activists working often in the background to protect species and bring them back from the brink of extinction. So this one is for the American burying beetle, and then Bob Mers from the St. Louis Zoo is depicted here. I believe he's on this call. I'm really excited to hear from his colleague Kayla later today. And I really wanted to highlight this as I think many of the folks who are going to be presenting today are often, working in the background and doing the day-to-day work to protect these species and bring them back from the brink. And we really want to bring that into the light. And yeah, with that, I will pass it over to Lou.
Thank you, Malia, and thank the center for having us and being able to share this amazing species with everybody. Next slide, please. So this is today's subject, the American burying beetle. It's the largest of the Nicrophorus species in North America, of 35 species, and the only one to experience a major decline. Next slide, please. Sexually dimorphic by the little marking above the mandible. You see on the left a little rectangle, it signifies male. The little triangle on the right is a female, so it's really easy for us to be able to distinguish sex. Next. So it is a carrion beetle, so it requires a carrion item as a reproductive resource, which is key to its life cycle. Male and female that win rights to the carcass will have that thing buried. In this case, it's a rat within 24 hours. Within 7 days, all fur and feather are taken off. It's rolled into this brood ball, secreted with anal and oral secretions that retard fungal and bacterial growth. The parents actually tend the carcass through the brooding process. Larvae go through 3 instars, and in the first instar, we'll stroke the mandibles of the adults to solicit feedings, which makes it a very unique species in that right. Next slide, please. So it has declined from 90% of its former range. It used to range from the central US all the way out to the East Coast, 35 states. The only place you can find it in the East now is a little island off the coast of Rhode Island, Block Island. That's our only extant population. And then there's 6 populations just east of the Rocky Mountains, South Dakota, Nebraska, Oklahoma, and Arkansas. Next. Next slide. Yep. So reasons for its decline, of course, habitat loss and fragmentation. This is, all the same suspected reasons you see for a lot of species that are endangered. Once you lose habitat, fragment habitat, you change species composition of that habitat, you push your predators out, you get more scavengers and mesopredators, and then you lose species, And this decline of this beetle was mapped with the decline and the loss of the passenger pigeon and other species like heath hens, Carolina parakeets that would have had high mortality events that this beetle would have cleaned up the mortality of and utilized as a reproductive resource. So now you have less resource, more competition for the resource. You throw in modern day stressors like pesticide use, nighttime lighting, and you have loss of 90% of its range. Next, please. So it was listed by the United States Fish and Wildlife as endangered in 1989, and with any endangered species, a recovery plan is drafted. And for this one, like many, it called for monitoring the existing populations, maintain captive populations in case of any stochastic events across the range, look to see an area, and they chose a small island off of New England to do a pilot introduction to see if it would be possible to release captive-born beetles successfully. And then we wanted to prioritize areas and conduct surveys looking for maybe additional populations across the range that have gone unnoticed. And then we wanted to look for other areas across the range to conduct additional reintroductions and manage new populations. Next, please. So our history here is we started in 1994. We got our first colony from the U.S. Fish and Wildlife soon after the pilot reintroduction on Penakees Island. We've produced quite a lot of beetles here. We've put 4,000 beetles on Nantucket Island since 1995. It was the first insect SSP for the Association of Zoos and Aquariums, but now it has been, I think, upgraded to a SAFE program, which we'll talk a bit more about later. Next slide, please. So I'm not going to go too much into rearing them because Mandy does a great job with that later on, but they're easy to house. They're inexpensive. It's great. Invertebrate conservation is great for small zoos or, institutions with small budgets. We house them in sandwich boxes. Next slide, please. We burying beetles, so we breed them in 3-gallon planter buckets filled with soil, and we provide them with a quail or a rat. Next slide, please. And this is our study site, Southern New England. And that island on the lower left is Block Island. That's where the extant population is. And then the island in the far left is Nantucket Island. And that's our release site. They had been seen there since 1926. We did extensive surveying before we released to make sure they weren't there. Next slide, please. So when we release them, we don't just say fly, be free. We actually pair them up. We bring them over in pairs. We dig a hole. We stick a quail in. We stick a predetermined pair based on genetics in the hole. We cover it with a turf plug, screen it off. Next slide, please. And this is our study site here on Nantucket. We're working on the eastern half of the island. We have trap grids and release sites on the eastern half of the island. Next slide, please. And in the monitor for them, we use pitfall traps, and methodologies are pretty much similar across the range. You'll see that with the other speakers today. We use wide mouth mason jars with a bait cup, baited with rotten chicken, and believe it or not, they come. And we have transect all over the island with 5 to 10 traps per site. Every beetle we catch is marked. Next slide. This is the pitfall trap. Once it's closed up, we do use foil covers, keep rain and sunlight out. Next slide, please. And again, every beetle is weighed. Next slide. Measured and marked. We usually use a cautery tool, and we have different notch schemes for different sites, or we'll use numbered B tags, and we keep track of all temperature and humidity data, not only at trap sites, but at the provisioning sites where we're actually pairing the beetles up with carrion. Next slide, please. And we've been keeping track for 31 years of all the other Nigrophorus and sylphid species on the island to see if we can, we're noticing any trends with them now that we've put this larger carrion beetle back into the landscape and all the meat we need to put in the landscape to keep that beetle on the landscape. Next slide. And then once we're done with all of our trapping, we pair up all the wild caught beetles and we provision them just like we did the zoo bred beetles at the site they were captured to bolster population. Next, please. And then we go back in 11 days and dig up 30% of the broods to kind of get our average brood size numbers for that year and what we think our F1 population is going to be. Next slide. And this is what we hope to find. We want to find mama still tending the brood and a nice healthy brood of third instar larvae on there. This is a sign of success for sure. Next. And then we go back in late summer, 45 days is pupation time for the burying beetle. So once those larvae, we count 45 days out from that larval count, and then we trap for F1 beetles, mark as many as we can, and that gives us what our overwinter survivorship is the following year. Next, please. So in 2011, we stepped back. We stopped putting out captive bred beetles. We provisioned only very few, and we did see a decline in the population. And we needed to determine, do we have a self-sustaining population? And is there enough suitable, available natural carrion on the island to support the population? So we did see a drop in the population, but we feel Nantucket's really important to have as an insurance colony in case something stochastic happens on Block Island. So we began to, in 2015, stop pumping as many beetles from the breeding program as we could back onto the island, provision every single wild beetle we get our hands on. Next slide, please. And now we have the beetles, again, thriving and distributed all over the eastern half of the island. We're really excited. We had great numbers in 2024. Next slide. And in this year, we broke all records since the 32-year history of the project, 31-year history of the project, based on trap nights. We've got the population back stronger and better than we ever had. So we're really excited about that. Next slide. It is Rhode Island's official state insect, thanks to a third grade class that lobbied a legislator to back the bill. Went to the state house, did a little PowerPoint about the beetle to the House and Senate, and here's our former governor signing the bill. And then, best of all, next slide. The beetle was brought to international attention by Dr. Jane Goodall, R.I.P. Jane. We're all going to miss you. You were a hero for conservation. But she wrote a whole chapter about this amazing species in a book, A Hope for Animals in Their World. I truly recommend you pick that book up. It's wonderful. Next slide, please. So with that, I'm going to hand it over to my colleague, Kayla, to talk about what's going on out west. Thank you all.
Hi, everyone. Thank you, Lou, for that introduction. I'm excited to talk to you all today about what recovery efforts for the American burying beetle look like in Missouri. We are the westernmost project right now working on recovery efforts reintroduction. So with that, next slide. So I just wanted to give you all a little bit of background about how we became involved in this project. The insectarium, or the invertebrate department, is one of the newer animal departments at the St. Louis Zoo. We officially opened our doors to our facility in 2001. And at the time, our curator was tasked by our new president to pick up a conservation project. That was something that was really important to our president at the time, that the zoo expand their efforts in active conservation work, both in the field and within the zoo. And so it happened rather serendipitously. We had someone bring in a carrion beetle and wanting to know what it was. Obviously, they're pretty distinctive looking. Those black and orange markings are very striking. We were doing research. We figured out, what it was. It was a burying beetle. But then we learned that there was actually an endangered species of burying beetle. And even more interestingly, that it was once formerly found in the state of Missouri. So that kind of got our wheels turning, and we started exploring what it might look like to get involved with what we then also found out was an ongoing recovery project that had already been started in certain other zoos, Roger Williams Park and Cincinnati. And so we decided to take this on. Next slide. The first thing we had to do, though, is determine whether or not there were any ABBs left in the state. Now, we truly thought that we would find them. We figured that they just weren't being surveyed thoroughly enough, that they had to be somewhere in the state. We are on the westernmost edge of the known populations that still are persisting in the wild. At first, we started to survey sort of where we could, but then we got more organized. We got more funding. Our program became a little more established within the zoo, and we knew that the last known specimen from the state was from the 1970s. They had not been seen since, as far as we were aware. We ended up surveying for a number of years, found tens of thousands of other Microphorus, but not a single ABB. So that gave us a certain level of certainty that, yeah, they probably were in fact extirpated from the state. Now, Western Missouri, it's a lot of tall grass prairie, wild habitat, as well as ranch land from the farming side. Next slide. It looks pretty different from some of the other reintroduction areas. It's a beautiful place. Western Missouri, some of these remnant tall grass prairies are absolutely stunning. I look forward to my opportunity doing field work every year. And it ends up being, it was actually also believed to be a good place for burying beetles, given that we were finding so many of the related species. Next slide. So we had to go through a few other steps. So once we knew that there probably weren't any burying beetles in the state, our next goal was to start our own breeding colony. Lou mentioned some of what they do, and Mandy will go into a lot more detail about what that looks like. But I did want to point out that our founder population is from a different location than the others. We got our original beetles from the population in Fort Chaffee, Arkansas. That was kind of the closest to where we would have where the naturally occurring population was closest to our eventual future reintroduction site. And over the past 20 years, we've had great success and have bred over 15,000 beetles. Next. So I mentioned there were other reintroduction projects going on, and in fact, we assisted with the efforts that were being done in Ohio between the years of 2005 and 2012. We were able to send staff, we were able to contribute beetles. We contributed beetles to other breeding colonies during this time, and we learned a lot from that. That was really instrumental for us to feel confident about moving forward. Next. So what we needed to do in order to begin releases in Missouri was we engaged obviously with the U.S. Fish and Wildlife Service to get a special designation for us to be able to release beetles within what we call a non-essential experimental population. It's a 4-county area in Southwest Missouri. Had to let folks know what was going on, and what was really important and successful in terms of getting this approved was that designation. Basically what that means is that private landowners could still do with their land what they were doing previously, so farming, ranching, things like that, without worrying about disrupting the beetles or the beetles disrupting those activities. Next slide. So our very first field site was Waconta Prairie in the town of El Dorado Springs. We actually released beetles there for 6 years at 3 different sites, continued to survey throughout the summer. We have 2 field technicians that we hire every year to survey throughout the area. And we also work very closely in partnership with the Missouri Department of Conservation, as well as the Nature Conservancy, which co-owns Waconta Prairie with MDC. And even work with them on projects such as learning how to assess the populations by a mark recapture project that we did with Missouri Department of Conservation for 3 years, which is why that beetle has a little tag on it, helps us know who's who. Next slide. After releasing for 6 years at Waconta, as part of our broader plan to establish a larger kind of meta population across the landscape, we started reintroducing at our second location, Taborville Prairie, including in 2020, which was a real achievement for us given the constraints placed upon us by the COVID pandemic. It was a very long, very hot day for the folks that did the release that year, because it was all done in one day. We didn't stay at a hotel, we didn't do anything. We drove straight 4 hours to St. Louis and then 4 hours back. Also continuing to survey, except for 2020. Next slide. So this is obviously a huge group effort. We couldn't do this without the support of many different partners. Not just St. Louis, but other zoos as well have contributed to our releases. Sedgwick County Zoo, the Wonders of Wildlife in Springfield, teens from our own zoo that have volunteered, as well as folks that came to us from other institutions. There's one on the far left, we have someone that came to us from the wilds and had brought her own experience working with burying beetles to the project, which was great. Next slide. So just to give you a very quick look at what it looks like to survey in Southwest Missouri, this is one of our traps. I don't know if there's audio, it does, but basically what you do when you check these traps is obviously they're baited with a bait, which in our case is rotting chicken. They can get really full. The numbers of sylphids in these areas are incredible to see. So I just wanted to share a little glimpse of what that looked like. Next slide. So 2025, this is a big year for us because this is the first year since 2012 that we have actually not done a release. We are at that point where we need to see what the beetles do on their own without our active intervention. So we'll be continuing to survey for the next few years to track that population, and that will really help guide our future decisions that we make. It was a very wet spring this year, so I wanted to include a picture of what it looks like to be driving around the prairie. There's a lot of mud. We got the truck stuck a couple of times, but we were good. We got it unstuck both times. Pretty proud of that too. Next slide. So yeah, we'll be seeing what our survey results, what in terms we get of our survey results. Also looking into doing more research to understand what conditions in the western edge of their range is most important for these beetles, what carrion are their larvae being raised upon, and whether or not we might even explore some future reintroduction locations in the future. And with that, I think I am turning it over to Mandy.
Awesome. Thank you, Kayla. So hi, everyone. My name is Mandy Pritchard. I am the team leader at the World of the Insect at the Cincinnati Zoo and Botanical Garden. Our insectarium opened in 1978, actually the first of its kind in the US. But I joined the team back in 2010 and quickly fell in love with the American Burying Beetle program. Since then, I've been working with the other organizations that are here today on this webinar to help bring this species back to Ohio. So I'm kind of in the center of the range, more or less. Today, I'm going to briefly just go over the history of this species at our zoo and then dig into what it takes to care for these animals, or what we call husbandry here at the zoo. Things like daily care, feeding, cleaning, and most importantly, of course, breeding. Next slide, please. So our zoo first acquired ABBs from Boston University back in 1991. They did a wonderful job breeding them for a few years until the colony eventually collapsed. That's likely due to inbreeding depression or nutrition, something we'll discuss in a few minutes. So our zoo's interest waned in the mid-90s, and we didn't work directly with the species again until 2009, when we reacquired them from OSU. Flash forward to more recent history, where we established a release site at a nearby preserve called Fernald Preserve, and held reintroductions there for a number of years until, of course, COVID messed everything up for us. But at that point, we decided to join forces more directly and more closely with the wilds, and we contributed our beetles from that point forward to their reintroductions. Next slide, please. So to breed beetles, you have to have beetles. So since 2013, we have worked with the Ohio Division of Natural Resources, the U.S. Fish and Wildlife Service, and Nebraska Game and Parks to acquire founders from the Sandhills region of Nebraska. Specifically, we source them from Rock County, Nebraska, which is in the center of this naturally occurring population within that state. And each year we are permitted to take up to 30 pairs. So we're very fortunate that we are allowed to do so. Next slide, please. Much like the others have mentioned, we also perform pitfall trapping in order to catch these founders. We use 5-gallon buckets, baited, of course, with carrion. And on a good night, we will catch a couple hundred beetles in just a few traps. We then drive these beetles back to Ohio, where they are split between the wilds and the Cincinnati Zoo. And I want to mention, too, that the wilds accompanies us and is along for the entire process of capturing these beetles in Nebraska as well. Next slide, please. So once they are back at our breeding facility, they are set up to breed using 5-gallon buckets. We use a lot of 5-gallon buckets in this program that are filled with soil. Each pair of beetles is provisioned, of course, with a rat carcass. We usually pick one that weighs around 200 grams. The beetles mate, and then they work together to bury the carcass overnight, sometimes all the way to the bottom of the bucket. It's really impressive. And they kind of get under the carcass and excavate soil out from under it until it just sort of sinks into the ground. Nature's undertakers, we call them. While they bury the carcass, they are also, oh, I'm sorry, next slide, please. So while they bury the carcass, they are also ripping off the fur and covering the carcass in antimicrobial secretions that preserve the carcass for their future offspring. Lou talked a lot about this as well, so we're just gonna refresh you. I refer to the brood ball, as Lou more professionally called it, as a rat meatball, because at this point, other than the tail, you can't really tell what it used to be. The whole feat, though, is very impressive, and if you consider the size of the beetle compared to the size of the rat, it would be the equivalent of you and your partner burying an elephant overnight without tools or secretions. Next slide, please. So after about 10 to 14 days, we will excavate that carcass from the bucket, or what's left of it, and we will count how many larvae they produced. We have had as few as 1 larva in a bucket, and we have had as many as 56 in a single bucket. I consider a good average, though, to be about 25. After the carcass is consumed, the larvae will wander off into the surrounding soil in that 5-gallon bucket and undergo pupation, which takes roughly 6 weeks. Next slide, please. And then about 2 months after the entire process began, you get brand new beetles emerging from the buckets. This video was captured using an infrared camera set up over one of our buckets this past year. I'd never done this before. It was really cool to see them emerge throughout the night without having to actually stay up all night. In the center, you can see there's a dish we actually added, and we filled it with live mealworms for the beetles to eat while they were waiting for us to come in the morning and process them. And they are very hungry at this stage, so all of the mealworms were gone by the time we got in the next day. Next slide, please. So speaking of processing these beetles, at this point we weigh and measure each individual beetle, and we set them up in their own containers with a label telling us exactly who they are, who their parents were, or even their grandparents, if they're a late enough generation, what they weigh, any special notes about them. All of this data is also stored in spreadsheets, and we track the individuals this way because inbreeding this species has historically been the downfall of many captive breeding populations. So we have to keep them separate, and it takes up a lot of shelf space eventually. So after processing, the beetles are then fed, and their habitats are cleaned twice a week. So feeding and cleaning happens twice a week for each individual beetle. This is no big deal when you have 50 beetles, but it becomes extremely time consuming when you have 500 beetles to care for. So this is not a one-woman show. I have a lot of help over here. The adult beetles are primarily fed live prey at our facility and the others. Historically, it was mealworms and waxworms because they're easy to get a hold of. However, in 2023, my team started questioning this diet, wondering if we could do better. So my coworker Amanda and I started trialing other foods and narrowed it down to a few things that they seem to prefer. Next slide, please. So then Janelle, my colleague from the wilds, who's going to speak next, sent us this paper that talks about a nutrition experiment done with Necrophorus orbiculus, so same genus, different species. It's a close cousin of the ABB. In this study, they found that the females that were fed blowfly maggots developed ovaries that were twice the size of their mealworm-fed counterparts, had far more offspring, and developed more juvenile hormone than beetles kept the old-fashioned way on mealworm-only diet. So we decided to mimic this experiment. We ended up raising flesh fly maggots, which isn't the same fly, but it was easy to get a hold of, and the maggots themselves were larger. And since ABBs are larger than orbiculus, we also thought that would be a good thing. Long story short, the ABBs devoured them. They could not get enough of them. You can see in this video, they're actually eating in broad daylight, which is unusual because they are a nocturnal species, and you can see the mealworm in the background being ignored for the maggot in the beetle's mandibles. Next slide, please. So for reference, this is a depiction of our breeding success in terms of average larva per successful brood prior to feeding out maggots, right? So this is before the maggot experiment. You can see the purple bars in the background indicate how successful the parents that we captured in Nebraska are compared to the average brood size of the F1 parents, the first generation parents, in pink in the foreground. And you can see how steep the decline is between one generation to the next. Also, we have a near perfect success rate with wild-caught parents at 96%. Every once in a while, we'll get one that's a dud, compared to a 50% success rate with the F1 parents, which is pretty terrible by comparison. So that means that over half of the buckets, or about half the buckets that we set up with F1 parents failed to even bury the rat, prep it, lay eggs, any of it. Next slide, please. And then on this side, which is even harder to look at, sorry, I added how our numbers look after feeding out maggots to our beetles. And you can see that our average brood size has skyrocketed, and some generations are even higher than wild-caught parents. Additionally, our success rate has jumped back up to about 85%, which is astounding. And so because of this, we are able to actually breed more generations per year and provide even more beetles for reintroduction. So suffice it to say that ABBs really like to eat maggots, which seems obvious to me now that an animal predisposed to locating carrion would relish eating maggots, but hindsight is 2020. Next slide, please. So because of this ABB baby boom, we have decided that we can now support an additional reintroduction site. So just this last summer, we did some baseline surveys at a nearby property owned by the Green Acres Foundation near Cincinnati, and we are slated to hold our first reintroduction there next summer. And we are excited to see how that goes. And we will continue to contribute beetles to the reintroductions at the wilds as well. That group picture up there was our huge volunteer group that came out to the reintroduction at the wilds this year, which Janelle is going to talk more about their program up there. So thank you all very much. And with that, I will hand it over to Janelle, who will talk about the wilds. Thank you.
Thanks, Mandy. Hopefully everybody can hear me okay. And here I'm in the beetle barn. As you can see, it's got lovely beetles behind us, or at least they're containers. My name's Janelle Urig. I am the Director of Ecology here at the Wilds. And I've been in this position for just about 5 years and been at the Wilds for almost 6 years. Next slide. So since everybody has covered a lot of the basics about the ABBs, I'm just going to talk a little bit about our program and some of the exciting things that we have going on. So just briefly, 2007 is when we began breeding. So our breeding barn, our beetle barn was finished, and we began getting some beetles from OSU and started breeding those beetles. By 2010, though, the colony collapsed, and that pushed us to get new founders from the Arkansas population and St. Louis Zoo. So we're trying out new genetics. The reason that we were theorizing that maybe the population crashed was weather-based, or we thought at least it would be a better option to get some beetles that were from down south and were kind of a little bit newer generations. So we continued to breed those in captivity. And then it wasn't until 2011 that we actually had our first release here at the Wilds. So at the time, there was other places that were releases were happening in Ohio. So OSU and the Cincinnati Zoo have been involved for a very long time. And so there were several sites prior to 2011 where beetles were being released. But the switch to the Wilds was, I think, a key one because we could release on site at our property. So it was a little less red tape to kind of cut through. And also we just have a ton of land here. So we got almost 10,000 acres, and then it's surrounded by more acreage. So, and then in 2013, we received new founders from Nebraska. So again, we were having some troubles with the colony, wanted to try out a different wild population, and then really began working with the Cincinnati Zoo. So sharing beetles with them just to increase genetic diversity. 2017, we had our first ABB capture. So 2017 was a really key year because the amount of effort that went into trapping drastically increased. So we went from trapping to kind of assess the population and assess overwinter beetles for a few weeks in the spring and a few weeks in the fall after we did releases. And we didn't have any success, didn't catch any beetles in those traps. 2017, with the effort of one of our previous employees, Andrea Malik, she was in that picture that Kayla had shown, she was able to really put in the effort, check traps pretty much from spring all through summer and into the fall. And so we had our first capture in 2017. We had more traps out, and we had higher effort on that. 2019, 2 years later, is when we got our first overwintered beetle. So that was really exciting. We know that now the beetles can survive over winter, and that was a big question that we were unsure of, especially at the Wilds where our soils aren't the greatest. They're compact, and they need to get below that frost line to overwinter. 2021, after 2020, we really shifted to releasing only at the Wilds, and so Cincinnati Zoo started releasing here. And that really helped to amp up our efforts in how many beetles we had put out. 2025 has been a record-breaking year, so I'll talk a little bit more about that in a moment. And then 2026 will be exploring new partnerships Mandy had talked about, and also some old partnerships coming back into the scene. So OSU is going to be working with us once again. So next. So I just wanted to touch on a little bit about what we had been experiencing here at the Wilds. So this is our typical setup for releases. Others have touched on more of the nuances of it. But basically, after we've released pairs, 2 weeks later, we'll go and we will check the brood chambers and see how many larvae we have, see which ones were successful, and then we'll have an idea of relatively how many beetles we might see in our traps later in the fall when they come up after metamorphosing. And so we had been doing a percentage. That was the common thing. Unfortunately, my first summer, full season, we were checking a transect and I had 0% success with the first kind of group that we checked. So I was like, oh, let's check a few more. Let's check a few more. Ended up checking the entire transect. And so after checking the entire transect, we did have success, but it really pushed me to think, maybe we should, check every single brood chamber. And upon doing that, we were able to find out that our released beetles were also experiencing that reproductive decline. And so only about 15 to 17% of the beetles that we were releasing, pairs that we were releasing, were successful. And so that's really where Mandy's study came into play, and is also really important because the more beetles we can get out there, put out there, the more likely a population will actually be sustainable. And so since then, we've reverted back to only checking a percentage, because now we're also seeing that success in the beetles or the larvae that are produced. So not only did we see that in our captive beetles, now that we have the nutrition down, we've also seen the increases in those larval checks. And so we went from 15 to 17% to about 30% just this past year. So doubled our success rate, and hopefully it'll just keep climbing up and up. Next. And also trapping. So trapping, like I mentioned, we didn't catch our first one until 2017. Since 2017, we've really kept our efforts in trapping every single day, or at least every single weekday. This has really given us a great idea of when beetles are coming up, what times of the year they're coming up, temperatures. We record temperatures every day that we check the traps. Also rainfall, looking at some different factors that might influence beetles emerging. And so with the trapping, we were pretty steady. It went relatively close to basically how many we released. So if we released a small amount, we would have a small number of beetles that we would catch in our traps. If we released a large amount, then we would have a large amount of beetles in our traps. And so this year, with those increases, we also saw that increase in the number of beetles that we trapped this year. So our record was 108 captures, and we actually tied that record this year. The big difference being that record, 108 previously, there were much fewer unique individuals and more recaptures, whereas this year we had a lot more unique individuals and fewer recaptures. So a lot more beetles getting out there, and we're actually getting them in our traps. Now, these guys can fly, obviously, so they can just go off the property. They've got a lot of area all around us. And so, there's chances that we're just not catching everybody. But the fact that they're going through metamorphosis successfully and kind of tracking with the trends is really exciting. Next, please. And so, yeah, just 2025, that record year. So it's the most beetles that we've released to date. So just last year, we released 1,104 beetles. That contained 348 pairs, 408 individuals. And then also, like I mentioned, we had those 108 captures, and then our 30% success rate with our released beetles. So things are going great and only getting better. Next, please. And so I just wanted to touch real briefly on what are things that you guys can do to help. So planting native plants in your backyard or on your property, that provides the habitat and food for native insects like American burying beetles. If you do see an American burying beetle, especially in Ohio, but also in other areas, in other states, reach out to the Natural Resources Department, because I'm sure they want to know. Also, you can also contact one of us at the zoos. We can also help confirm whether or not it's an actual ABB. But like I said, they can fly anywhere. So we want to see that they're expanding and surviving outside of the wilds. And then lastly, reducing pesticide use in your gardens and lawns, and also reducing rodenticide use. So, you know, the thing that the ABBs are looking for most are those dead rodents, and some of those might be suitable enough to be carrion for the ABBs. So that's my plug for what you can do to help. And then Lou's gonna kind of close out the talk with some exciting news.
So yes, the Association of Zoos and Aquariums, which is the accrediting body for all of the zoos involved in the call here today, has a program called SAFE, which is Saving Animals from Extinction. They are usually all about very threatened and endangered species, and we've just found out this June that the American burying beetle was approved to be the 54th safe species. So what that means is all of my colleagues on this call today, we're all going to be working together. We're going to draft a 3-year plan. It's going to allow us to standardize husbandry and field techniques range wide. You know, the diet stuff Mandy's working on. Help again continue to support release initiatives and explore and help fund, because this allows us now to be eligible for a lot more grant opportunities and to be able to get contributions from supporting institutions to help support some of our priority initiatives for research, like stable isotope work, to see what these beetles are utilizing in the landscape, genetics across the range, of course, effects of climate change, and again, anything that would help with the captive breeding of the species and its conservation in the wild. We're going to all work together to continue to support these releases we all talked about today, and continue to hopefully use community science initiatives to conduct surveys in areas within the historic range that the beetle no longer exists, to get kids out in the landscape and really develop a great outreach and education component that anybody can use to celebrate this species. And then just you know, continue to work together to keep this beetle on the landscape. And this is going to be a great way for us to do it with great accountability and, again, support from not only our holding institutions, but institutions that aren't holding the beetle, that can help us spread the story and fund some of this amazing work. So thank you guys for joining with us and having us on the this webinar.
Thank you. Yeah, thank you to each one of you. And this was fantastic. I'm so excited that this all came together, and we have quite a few questions already in the Q&A, so I can kind of get us started there. The first question is actually from Noah Greenwald, who is the director of our endangered species program at the center. This is for Lou. Why do you think that the Nantucket population may not have been self-sustaining? Any insight there?
We think I mean, this thing would, I mean, it's not really a habitat. It's resource dependent. So it needs to find that suitably sized carrion that hasn't been taken over by flies or ants. So we're just, you know, we're questioning, is there enough suitable size available carrion on the island to support the population? But we have done some preliminary stable isotope work, both on Nantucket and on the extant population on Block Island, because we always wondered, why did they hang on Block Island, and we're seeing them dive on Nantucket, and it was always thought to be ring-necked pheasant. There's a naturalized population of pheasant on Block Island, and through isotopes we did determine that yes, they are utilizing pheasant. But we also found that they're utilizing things on different sides of the weight range. They're utilizing smaller things, just not producing larger broods, and using larger species. We had a brood, instead of we provisioned it on a city pigeon, which is like 500 to 700 grams. Rather than preparing the pigeon, they hollowed out the breast cavity and raised the brood inside of the bird. And we've seen blackback gulls where way too big, but they buried the head and raised their brood on the head. So we're hoping that, you know, we just didn't have the carrying capacity out there. So we're going to pump beetles back in the landscape, plus our partners on the island, Nantucket Conservation, and you know, I think Kayla had mentioned that this takes a village, and you know, we have everybody from U.S. Fish and Wildlife, Nature Conservancy, Nantucket Conservation. They're doing a lot of work out there for habitat restorations, and we're seeing species out there now that we never did see before. So we hope that, you know, we're gonna see that curve going back up. But very good question, and we ask that to ourselves every day. So I hope that answered your question.
Thanks, Lou. And I think there are a couple other questions that sort of are interrelated, so I'm going to kind of pull them together, and if this doesn't cover the questions, you know, feel free to ask again. But someone asked, so when we reintroduce the American burying beetle, do we also restore former natural habitat and other species of the native ecosystem? And sort of relatedly, are there observable changes in the introduction sites themselves since the burying beetles have been released? That's for anyone who wants to jump in there.
So I can speak from the St. Louis perspective. For your first question, do we also restore habitat as part of our ongoing projects? So we don't necessarily actively participate in the restoration of these sites. In part, we had chose the sites we did because they were already fairly protected and had that available carrion base as well as evidence of those similar species of beetles to suggest that they would support the American burying beetle. But honestly, I think it's a great idea. And there's a lot we still don't know about how certain habitats and for example, in the West, we have a lot of, wildfires, or we would have had wildfires historically. And the prairies that we do reintroduce our beetles on do undergo controlled burns at specific times of the year. So I think that's a wonderful thing. And I agree that, you know, you need that habitat in order to support these populations. But to Lou's point, even more so than habitat, it's the carrion. So having those habitats support the animals that will become the carrion that the burying beetles could then feed upon is really key. And then as far as your second question, which I'll have to be reminded what exactly that was.
Yeah, it was, are there changes, observable changes to the sites where you're releasing?
Right. No, that's an excellent question. And that's not necessarily something that we have noticed on our sites. But that's a question that we have begun to kind of think about what's the best way to explore that. Like Lou said, we do track the populations of the other carrying species on our sites through our survey efforts. So we have the data that goes back many years that kind of looks at those populations. However, these are naturally fluctuate a lot, and a lot is dependent on weather. It's dependent on overwintering conditions. So I can't say with certainty what impact reintroducing American burying beetles has had on these landscapes, but you know, it does introduce a potential for competition because a lot of these burying beetles are utilizing carrion, although it might be a different size carrion, or they raise their brood at a different part of the year. So there's a sort of like a time, it's spatially or temporally separate. But you know, I think there probably is some impact. We just might, I'm speaking for St. Louis, not understand the full details yet.
And just because you touched on this, there were a couple other questions about how do burying beetles, because you have mentioned that you found several different species of burying beetles, how do they share provisions or share habitat with other burying beetles? Like, are they coexisting? It sounds like you're describing that they might be active at different times of year, things like that. And then there was sort of a secondary question, what makes the American burying beetle seemingly more vulnerable comparatively?
I think the burying beetle is more vulnerable because it's more of a specialist as to carrion size. They don't have much of a size range, where some of the other species, and like we said, some are diurnal, some are nocturnal, some are more active in the late summer than the other summer. But the American burying beetle has a short window of activity, and we have noticed on the islands through overwintering mark recapture studies that 75% of them die naturally over winter. So only 25% of that population survives the winter. And so they really need to recruit and find carrion to keep those populations going. So it's a species that could crash hard really fast. And we had, just like, my colleagues had mentioned, COVID really killed us too, because we couldn't get out to the island for 2 years. And then we really started to see, without any supplementation at all, that population really going down on us. So, but it didn't take long to bring it back up. And, if they have the resource, and back to the habitat question for us on the island, we work on conservation lands that are managed for the conservation of many other species, which, like Kayla referred to, you have a suitable habitat for the rodents, the birds, and we, through isotopes, and notice they're using insectivorous birds, seed-eating birds, so they're utilizing things like towhees and robins, and so yeah, the more diverse the habitat, the better the chance the American burying beetle has to find that available carrion at that window of activity in the early summer.
Great, yeah. We had another question for everyone, or one person who wants to jump in. What types of certifications are required to do beetle husbandry? I'm sure that might get into the weeds, but if you have sort of a brief answer.
Go ahead,
Mandy?
Lou. I was just going to say that, I mean, I think for many of us, we exist at institutions in the first place, and then, you have to hold permits with the Fish and Wildlife Service or with your state organization, or both sometimes. So it's a lot of red tape from a permitting perspective, was all I was going to say. I don't know, Lou, if you wanted to chime in too.
No, I was going to say it's all about the permit, and showing that you have some level of experience with a Nigrophorus species, and we like to see some multi-generational breeding of at least one of the related species before we allow them to work with American burying beetles. And then yes, getting them on the permit. They need to be named on the permit. We just can't take any volunteer and let them go unless they're supervised by a person who is permitted to work with the species. But unfortunately, you're looking at all the people, most of the people that work with it. So it's going to be hard pressed to find, a student or a grad student or somebody who's had experience with this species, unless they've crossed paths with one of us or have worked with one of our partner universities like OSU or so. Yeah, but we'd love to train you. We need the next generation.
And I was going to add, if you're specifically interested in invertebrate husbandry, my background is as a zookeeper at St. Louis. So if you're interested on that part, you will naturally fall under those permits. If you were to come work for St. Louis as a zookeeper, all of our zookeepers work hands-on with the rearing and the care for the beetles on a daily basis.
Yes, as Lou said, we are looking at the dream team right here. We do have some more questions in the chat, but we are also at the hour, so I think I am going to end it here, because I know we could keep asking you questions until sunset. Just want to say just a huge thank you to each of you. It's been wonderful to put this webinar together, and just your wealth of experience and knowledge has been incredible to work with and here to see on this webinar. Here are some resources for all of our attendees. Thank you all for joining. If you have any questions or have any specific questions that didn't get answered, feel free to email me and I can direct your questions to the right person. And we will be sending out a follow-up with the webinar recording, which will also be posted on our YouTube video. Well, thank you all and have a great rest of your day.
Thank you. Thank everybody for joining.
Thanks for listening.
Thanks, everyone.